Good afternoon, ladies and gentlemen. On behalf of Manila House, I'm Bambina Olivares, the Director of Programming, and I'd like to welcome all our members and guests to this very timely webinar on the COVID-19 vaccines with Dr. Benjamin Ko, presented in partnership with CMI. CMI, if you haven't been yet, is the first premier VIP concierge clinic in the Philippines, and it's a safe space with no queues and no crowds, which is very important this time of COVID. And before we begin, I'd like to present a short video about CMI to give you a better idea of what the facilities are all about.
Good afternoon, everyone, and we hope you are well. Centre Medical International, or CMI, is a premier outpatient clinic located in the heart of BGC. With over 150 specialists and subspecialists on our roster, CMI continues to be here for your medical needs, such as specialist consultations, by phone, video, or in person, diagnostic procedures, and preventive treatments, such as vaccinations and checkups. No crowds, no queues, no hassle. CMI is the safe clinic for you and all those who are dear to you. CMI is open Mondays to Saturdays, 8 a.m. to 4 p.m. Visit our website www.cmi.care or visit our Facebook page facebook.com/cmi.care for the latest updates.
Thank you for that brief presentation. And now, before we begin, I'd like to call on one of the founders and directors of CMI, Dr. Hazel Zulig. She's a doctor herself, a neurosurgeon, and she will introduce today's speaker, Dr. Benjamin Ko. Dr. Zulig.
Thank you, Bambina, and welcome all to the Manila House webinar on vaccines. I am a proud Manila House member also, and it's nice to be on this forum with all of you. Let me introduce our speaker for today. He is one of the most sought after speakers in this time of COVID. Dr. Benjamin Ko is a pediatrician and a member of the Pediatric Infectious Disease Society of the Philippines. More importantly, for the topic for today, he is a clinical pharmacologist and the former director of the Center for Drug Regulation and Research of the FDA. That is precisely the office that gives authorization and approval to medicines and vaccines. So he is really the point person when it comes to discussions about vaccines. So CMI is very proud to have him in our roster. He was our first medical director, and now he is here gracing our Manila House event to talk about vaccines, truth or dare. Thank you.
Before we turn over to Dr. Ko, just some rules. After Dr. Ko's presentation, we'll be open for questions. So please put your questions in the chat box or in the Q&A box, and we'll get to them. We'll have time. I'm sure everybody has a lot of questions, and we'll make time to try to make time to address all of them. Thank you. Dr. Ko?
Hi, good afternoon, everyone. Let's get to the bottom of my talk this afternoon. Hopefully, it will be a fruitful afternoon for all. For some of you who have heard already my lecture, I have given this in the past. This is actually an updated version. So let's start with, well, why call it truth or dare? Well, because we'll give you the facts, and all people will always say, why don't you want to dare get a jab? Well, it's because a lot of us don't know a lot of information. So today I will talk about basic immunology. I know most of you may not be doctors, but for those that are not, this will help a lot, particularly when we talk about the concept of why and how vaccines work, and then how vaccines are produced, the development process, and where we are today with respect also to adverse events and the fear of vaccination. And of course, finally, we summarize everything. So let's talk about Immunology 101. And when we talk about Immunology 101, this is a cartoon slide, no? And I show you what the existing pathogens are. These are measles, chickenpox, and the flu. A pathogen is actually a bacterium, a virus. It's a microorganism that can cause disease within the body. And each of them are made up of actually several subunits or subparts. And it is unique to that particular microorganism and the disease it causes. For example, I drew it in ways where for measles, you see the sharp edges. For chickenpox, there are middle crosses. For flu, there are circles. And for a new pathogen, of course, there are little boxes. And the antibodies produced in response to the pathogen's antigen are important. They are important for the immune system because these subparts, these little parts that protrude from the cell, is called an antigen. So the antibodies that that are produced to respond to that particular antigen plays an important part in our immune system. When our body is exposed to an antigen for the first time, like in SARS-CoV-2, it takes time for the immune system to respond, and it takes time for the immune system to produce specific antibodies. In the meantime, what happens is that because we do not have any antibody to it, we get sick. And that's the reason why we get sick with SARS-CoV-2. So a vaccine contains actually a weakened portion of that antigen, no? Or it may be an inactive part of that particular microorganism. And it's that antigen that triggers the immune response within the body. Newer vaccines contain the blueprint for producing antigens rather than the antigen itself. And regardless of whether the vaccine is made up of the antigen itself or the blueprint, that weakened version will not cause disease, but it prompts our immune system to respond in the same way as it would have on its first reaction to getting exposed to the microorganism. So some of the vaccines will require multiple doses. They're even given in weeks or months apart. When the vaccines were initially rolled out for SARS-CoV-2 or for COVID-19 vaccines, they were spaced 3 weeks and 4 weeks apart. But now we know more information that some of them can be even spaced months apart, and some can be even given as a single dose. And this is needed to allow for the production of what we call longer living antibodies and the development of what we call memory cells. This way, our body is trained to fight the specific disease-causing organism, build up memories of the pathogen, so that if and when we get exposed to it in the future, we can rapidly fight that particular microorganism. And then, of course, there is the concept of herd immunity. A vaccine will always protect an individual, but there will be people whom we cannot vaccinate. And when someone is vaccinated, they're likely to be protected against the targeted disease. But not everyone can get vaccinated. And so people with underlying health conditions that weaken the immune system, those who have cancer, HIV, or on immunosuppressive agents, or have severe allergies to some of the vaccine components, may not be able to get vaccinated with certain vaccines. But remember, then a lot of people in a community are vaccinated. The pathogen has a hard time circulating because most of the people it encounters have become immune. That's the reason why more people should get immunized. The more people that get vaccinated, then the less likely people who are not able to get protected by the vaccines become at risk of even being exposed to the harmful pathogen. But no single vaccine provides 100% protection, and herd immunity does not provide full protection to those who cannot safely get vaccinated. With herd immunity, however, these people have what we call substantial protection because those around them are getting vaccinated. So when people around you are getting vaccinated, then there is a higher chance that they're not going to get sick. And if there's a higher chance that they're not going to get sick, then there's a higher chance that you're not going to get sick as well. But is herd immunity possible for COVID-19? And that's when I have a push against the term herd immunity because I think a more suitable term is herd protection. It's very difficult to demonstrate herd immunity as the government and the WHO and other health agencies tout herd immunity because we have to remember that herd immunity is only possible if humans are the final host, like in measles, in chickenpox. Unfortunately, um, for SARS-CoV-2, an animal reservoir is present. And we've known that from the studies where minks, cats, and other pets can get SARS-CoV-2. Studies will need to be done also later on for patients that are not immunized. And this will take much longer and more expensive to do. For example, if we have 100 million Filipinos and you say that 70% need to get immunized to get herd immunity, that means we need to immunize 70 million people. But you have to remember that in the Philippines, the average age is 23. That means that almost 50% of the people in the Philippines are below 18 years old. And we know the vaccine cannot be given to those 18 and below yet. So that's 50-50 again. So how many of the people are eligible for getting immunized? In reality, of the 100 million, let's say about 55 million are eligible to get immunized. Of the 55 million, then there is hesitancy in 50%. That means only 27 million will get immunized. And that is way below the 70% herd immunity. On the supposition that 70 million people do get immunized, the only way you can determine whether the vaccine works as herd immunity is possible is that the remaining 30 million Filipinos or the 30 million people will need to get swabbed to determine their antibody status or to determine whether they develop an infection or not. And it's kind of expensive to swab 30 million people after so many years. And current clinical trials do not address whether there is an interruption of transmission for SARS-CoV-2. So there are major limitations to all of the clinical trials that are going on. And you have to remember, there's a large asymptomatic population that gets infected with SARS-CoV-2. So you don't know if people who don't get sick have or don't have SARS-CoV-2. So let's go to the vital issues. The vital issue is one, the endpoints for clinical trials for COVID-19 vaccine. It's important for everyone to remember that all the clinical trials that are being done for all the vaccines globally have two endpoints. One, that they should demonstrate the first one is the prevention of PCR confirmed symptomatic COVID-19. What does that mean? It means that you don't get sick. You don't come down with the infection. But like I said, because there is an asymptomatic, patients can be asymptomatic, you may have received the vaccine, you do not have symptoms, but you are infected. Because there's a possibility that you get infected along the way, but you don't have any symptoms. So the first one is that patients should not get any symptoms at all. The second endpoint to me is the more important one. And this is the prevention of PCR confirmed symptomatic moderate or severe COVID, which means that the second endpoint is that those that do receive the vaccine will still get sick, but they will be mild and will not die. So between the two endpoints, the first one is you will not get COVID-19 at all. And the second endpoint is you will get, but it will be mild and you will not die. The second endpoint is the more important one. Now, how are vaccines developed? Well, basically, the key ingredient is the antigen, the one I explained to you a little while ago. And for COVID-19, for SARS-CoV-2, they're the little boxes that you see outside the shell of the cell. It's either a tiny part of the disease that causes of the disease-causing microorganism, or it can be a weakened, non-dangerous version so that your body can learn to fight it without getting sick. But that's not all what vaccines contain. I think to laymen, they may think that it's just an antigen. Unfortunately, all vaccines contain other things. Like any medicine, it needs to contain other substances in order for it to stay potent. So they are preservatives, stabilizers, surfactants, residuals, diluents, and adjuvants. And all of these things that come in together with the vaccine can cause an adverse event, not specifically to the antigen, but because of the other things that are added in a vaccine. Why do you need to put them? Well, because they serve a purpose. Preservatives, for example, prevent the vaccine from getting contaminated when the vial is opened, particularly if you are using a multi-dose vial. Sinovac's vaccine is a single dose vial, so they do not contain preservatives. On the other hand, the vaccine from Pfizer, the vaccine from Moderna, the vaccine from Astra Zeneca are multi-dose vaccines. And therefore, there are preservatives in it. As a matter of fact, because they are multi-dose vaccines, and it will the the concept of multi-dose vaccines is that it makes the vaccine cheaper. Why? Because glass is expensive. Okay? So everybody has to remember that. All multi-dose vaccines end up cheaper. When the government purchases for an expanded program on immunization for children in the community, the vaccine they buy are multi-dose. They do not purchase single-dose vaccines like you give in the clinics. Now, there are stabilizers. And what do stabilizers do? They prevent chemical reactions from occurring within the vaccine, and they keep the vaccine components from sticking to the vaccine vial. Otherwise, they will stick to glass. They can either be sugar, amino acids, gelatin, and other different proteins. And of course, there are what we call the remaining substances, such as surfactants, which prevent the settling and clumping of elements. There are residuals, which are tiny amounts of substances that are used during manufacturing. They are not active ingredients, and they are only present in parts per million or parts per billion. The diluent is the liquid that is used to dilute the vaccine. It's usually sterile water or sodium chloride. And of course, the adjuvant is the one that is there to improve the immune response in order to keep the vaccine at the injection site a bit longer, or in order to stimulate local immune cells. So most of the viral vector vaccines contain an adjuvant, including the inactivated vaccines. Why? Because it serves that extra kick to stimulate local immune cells. Now, all of the vaccines go through what we call a clinical trial. And some people say, well, it's dangerous, no? The first phase is a preclinical phase. In short, we do it first in animals in order to evaluate its safety and the potential to prevent disease. Even in animals, we try to find out if there is that kick, that immune response. Because if there is no immune response in animals, then there will be no immune response in human beings. Once we're able to demonstrate an immunologic response, then we can proceed to human clinical trials. And there are different phases of the clinical trials. In summary, the Phase 1 are in healthy human volunteers. Okay. While phase 2 expands now to determine the dose, the correct dose. And phase 3 expands further to a larger population in order to assess, again, safety and efficacy at that selected dose already. So for example, during the phase 2, we try to find out does 0.3 ml and 0.3 ml work, or 0.5 ml work, sorry. And if we find out that at 0.3 ml, the vaccine works the same way as 0.5 ml and has less side effects, then that means the one that you will launch is 0.3 ml because they are both the same in efficacy, but it is much safer to give 0.3 ml. So that's the one that's used now in the phase 3 clinical trials in order to demonstrate broader efficacy and more safety issues. And then it gets rolled out eventually. And the rollout is a review process by the different regulatory agencies. The more mature, we call it mature, regulatory agencies include the United States, Europe, and Japan. So they are mature regulatory agencies. And once the more mature regulatory agencies have approved a vaccine, then that means that most of these vaccines are ready for what we call EUA, or emergency use access, especially during a pandemic setting. The overarching goal is that it must be proven to be both safe, see the words, nauna ang safe parati kaysa sa effective, because you can have an effective vaccine, but if it is not safe for use, for human use, then it is useless in the end. And the bar for vaccine safety and efficacy is extremely high because we recognize that vaccines are given to people who are not sick and free from the disease. Finally, when it gets rolled out in this way, when it is approved by a regulatory agency for marketing, that means the drug receives a certificate of product registration, not just an EUA, but a CPR, it can be sold over the market already. And when I say sold, it can be commercially available, not just selling at cost like an EUA is. So phase 4 is one wherein it is rolled out completely. It is available commercially, but it is still monitored even after the vaccine is introduced so that we can still continue to monitor safety and effectiveness. We keep track of the vaccine's impact, and any data that is obtained will be used to adjust policies in order to optimize the impact and allow the vaccine to be safely tracked throughout its use. Once it is used, again, it should be continuously monitored to make sure it is safe. I'm sure if for those that are listening now, you will notice that as I go over the lecture, I keep talking about safety, safety, safety. So we are rest assured that everything that gets rolled out is safe. There is no drug or vaccine or any biologic agent that is 100% without any side effect. Even the measly paracetamol has a side effect. But one wherein the benefit will always outweigh its risk. And the journey of a vaccine begins with the cold chain. We always remember that in any part of the world, we must make sure that the cold chain process is well kept, from manufacturing from the development of clinical trials to its pre-qualification to manufacturing to cold storage to the delivery and to every vaccination campaign. So this is one whole journey of the vaccine. Once this is not, any part of this is interrupted, then you will get vaccines that will not be useful. It may not cause a side effect, but it will not, because it will not cause a side effect and it is useless, That in itself is a side effect altogether. The next is the concept of efficacy versus effectiveness. Because people always talk about this is clinically efficacious. Why is it clinically effective? There are two different terms. When we say efficacy, this is the degree to which a vaccine prevents the disease. And therefore, these are in clinical trials. So if I say a vaccine with an efficacy of 90% in a trial is seen, it means that there was a reduction in cases in the vaccinated group compared to the unvaccinated group. So one set of patients receives placebo or nothing at all, and the other set of patients receives the vaccine. So in those that received the vaccine, 90% of them did not get sick, while a certain percentage of those compared to the placebo got sick. But laboratory conditions do not always translate to effectiveness. So in an efficacy trial, you can overestimate the vaccine's impact in practice. Effectiveness refers to how it performs in the real world. In the real world, we do not have volunteers anymore. In the real world, we don't look at healthy volunteers and patients that are asked to not to get included in the clinical trial because of underlying illnesses. In the real world, everybody can receive the vaccine. So if the vaccine is effective, then the cases are more likely to appear in the unvaccinated individuals. So with that introduction, the next question is, where are we now with the COVID-19 vaccines? This is updated already for this talk. Currently, there are 37 in phase 1, 28 in phase 2, 20 in phase 3, 6 that are authorized, 4 have been approved for full use, and 4 have been abandoned. The abandoned vaccines include the one from the University of Queensland. Here, some of the volunteers were getting positive tests for HIV, even though they were not infected with the virus. The trouble appeared actually to lie in the vaccine design. If the spike proteins are not anchored to a coronavirus, they unfold, and the antibodies to that unfolded spike protein will not work against the folded proteins on the real viruses. So the researchers made a small change to the protein. They created a little clamp to one end to hold the molecule in its proper shape, but the clamp was similar to the protein that was on HIV, which can lead the immune system to make HIV-like antibodies. So, nagiging false positive ang HIV doon sa mga patients that received the vaccine, when in fact, they don't have HIV. So, this was abandoned by Australia. The other one is the Merck plus IAVI, because most of these vaccines are in collaboration with other companies. And this was based on vesicular stomatitis viruses. The design was designed as a pill, but they failed to trigger an immune system comparable to what happens in a natural infection with COVID-19. So they abandoned that vaccine. The other one is from Merck, together with Institut Pasteur and Themis, which are also vaccine makers. The vaccine used a weakened measles virus that carried the gene. The phase 1 trial started in August, and on January 25, they abandoned it because the vaccine provoked a response weaker than a natural infection. And the last one that was abandoned was the one from Imperial College London plus Morningside. It's a self-amplifying RNA vaccine. It's a beautiful vaccine. They collaborated with another pharma company on December 18. The pharma, I think, is called NSC, to formulate a solid version of the vaccine that is an implant on the skin without a needle. On January 27, they announced that it was not the right time to start a new efficacy trial for further vaccines, and they will turn their efforts to making candidates that will work well against the emerging variants of coronavirus. So you will notice that we got here in a warp, what we call warp speed. Of all of the vaccines, the first human trial came with Moderna. So Moderna was actually the first, not Pfizer, uh, to do the first launch of the vaccine, and that they began the clinical trial on March 16, the same time that we were on lockdown in the Philippines, in the US. And the Dutch study, you will notice that the the various studies came out on the occurrence of blood clots, on analysis concluding that 40 to 45% of patients get infected, are asymptomatic. So this is what I was saying, that there's a large asymptomatic phase. And in between these, a lot of the vaccines were actually coming into play. On August 11, the first country to approve a vaccine was Russia. That was their Gamaleya vaccine. This is Sputnik V. They approved it, but you will notice that the approval as an EUA in various countries came actually only in January, when it saw print in the Lancet. And Pfizer, Moderna, and Astra Zeneca all announced in November of last year a successful phase 3 clinical trial, which means that this is just 6 months, just barely 6 months into the vaccine development. And what data we know is that short-term safety is the rule. So let's go to how all of the 9, and I only put 9 because if we go through all of the vaccines that are in development, we will finish tomorrow. 9 important COVID-19 vaccines and how they work. I grouped them into different categories. Let's start with what we call the mRNA vaccines. There are 2 in the market. One is Pfizer and one is Moderna. Both of them have received EUA globally, but Moderna has not applied for emergency use access in the Philippines. So there is no application for Moderna yet. So there is no EUA for this. Only Pfizer has an EUA in the Philippines. The generic name is Tozinameran, and it is branded as Comirnaty. How do these mRNA vaccines work? They both of them are the same. They will work similarly. Interestingly, a lot of people will say, what does 1273 for Moderna mean? And interestingly, look at Moderna's name, mRNA for Moderna. So it's easier to remember Moderna as the pioneer for mRNA vaccines. And the 1273 means 1,273 amino acids in its chain. So that's something that at least now you know. And that's not given in every lecture. So what do mRNA vaccines do? This is the example of this is the figure of a coronavirus, no? A virus, you can see the spikes and of course the spike protein gene, which are found inside the cell of the virus. The spike proteins here that you see outside are the target for the potential vaccines and treatments. So both Moderna and Pfizer vaccine is based on the virus's genetic instructions for building these spike proteins. And mRNA is very fragile. So once you inject mRNA, it gets chopped into pieces by our natural enzymes if you give it directly into our body. So in order to protect the vaccine, the mRNA that you see in between here was ingeniously wrapped in a lipid nanoparticle that surrounds the mRNA. And the mRNA molecules actually quickly fall apart in room temperature. So this is not stable. That's why the Pfizer vaccines are more difficult to handle than the Moderna vaccines. Pfizer has now data that you don't need to store it at -70 or at ultra low temperature. And they have submitted, may I use my words correctly, submitted to the US FDA data that it can be stored at -15 to -25. Now, the stability data is still up for review. So, because it is still up for review, it does not mean that you can store it at -15 to -25 at once. Until the data is out, then it's still at -70. So when you inject the vaccine, it now bumps into the cell. Okay, so this is our vaccinated cell, and it fuses and releases that mRNA into the vaccinated cell. And then it produces a spike protein. 3 spike proteins make a spike, and these spikes and the protein fragments now try to insert themselves so that they can protrude outside of the cell. Some of the spike proteins form spikes that will migrate to the surface of the cell. It sticks out their tips, and the vaccinated cells break up some of these proteins into fragments which present on the surface. These protruding spikes and the spike protein fragments, the ones you see in the letter Y, are recognized by our immune system. So it enters our cell. Now our cell spots the intruder. When a vaccinated cell dies, so that particular cell that I showed you a while ago, the vaccinated cell, it's going to die. What happens when it dies? All of these now float in our body. Okay? So all the debris now float. And when a vaccinated cell dies, the debris will contain many spike proteins and protein fragments which are taken up by a type of immune cell called the antigen-presenting cell. We all have that in our body. The cell presents fragments of the spike protein on its surface, and there are helper T cells that recognize some of these spike protein fragments. These helper T cells eventually circulate in our body, and we have what we call B cells also in our body. These B cells bump into the coronavirus spikes on the surface of the vaccinated cell. Remember, we were talking about the vaccinated cell a while ago. Some of them won't burst. Some of them just are still there. So the B cells now bump into them. And when the B cells bump into the coronavirus spike, some are able to lock into the spike proteins. So when they are locking into the spike proteins, and if the B cells are activated by the helper T cells, these start to proliferate. And what happens if they lock into it is that they proliferate and they pour out all the antibodies that target the spike proteins. And all the antibodies that are now present in our system can latch on when the coronavirus is seen in our system. They latch on to the coronavirus. They mark the virus now for destruction and prevent infection by blocking these spikes from the virus from latching on to other cells. And that's the beauty of our antibodies. Again, those antigen-presenting cells can also activate another type of immune cells called activated killer cells in our body. They seek out, destroy the coronavirus infected cells that display those spike proteins and fragments on their surface. So that's the way the mRNA and most of the other vaccines work. The Pfizer-BioNtech vaccine requires 2 injections. The manufacturer says it is 3 weeks apart. The US FDA has said that it can be even given 6 weeks apart and has allowed it to be given for that prolonged duration of time. There is new data coming out of Pfizer that the first vaccine, after getting the first shot, there is already moderate protection, but you achieve best protection after getting the second dose, right? And you can see that in the chart here, where these are people that have taken the Pfizer-BioNtech vaccine and people taking placebo in the yellow line. For the yellow line, you will see the placebo patients. They did not get anything, so they just got infected over a period of time. While those people who got the Pfizer-BioNtech vaccine, there was an immune response. Some of the people still went on to getting COVID infections, but overall, after the second dose, it stopped. You see the same thing with Moderna's vaccine, except that the dose is 28 days apart and it is 0.5 ml 28 days apart. All right. Let's now go to the vector-based vaccine. These are non-replicating vector-based vaccines. And there are 3 in the market: Astra Zeneca, Johnson & Johnson, and of course, Sputnik V. Um, AstraZeneca is marketed as Covishield, while Gamaleya's product is marketed as Sputnik V or Gam COVID Vac. Now, how do the adenovirus-based vaccines work? ChAdOx1, otherwise chimpanzee adenovirus Oxford 1, nCoV-19, or AZD1222 is the generic name for the AstraZeneca vaccine. It is actually a DNA inside an adenovirus. The gene for the coronavirus is added to another virus. And here we have an adenovirus. The adenovirus typically causes colds or flu-like symptoms. It can enter cells, but it cannot replicate inside them. And this is not a new technology because we've used it for the development of Ebola, Zika, and HIV vaccines. The beauty of this is that the DNA is not as fragile as the RNA. It has a tougher protein coat that protects the genetic material inside. What happens when it enters a cell is that once you inject the adenovirus vaccines, it enters the cell the same way as it did with the mRNA. The adenovirus bumps into the cell. It latches on the protein of the surface. The cell, our cells engulf the virus in a bubble and it pulls it inside. Once it is inside, the adenovirus now escapes from the bubble. And once it leaves the bubble, it actually attaches itself to the nucleus. Once it has attached itself to the nucleus, the adenovirus now pushes the DNA. See the DNA inside here. It injects it now, pushes it into the nucleus, and it is engineered. It is a recombinant vaccine. It is engineered so that it cannot make copies of itself, but the gene for the coronavirus spike protein can be read by the cell, and it is copied into a molecule called mRNA. And the whole process on how the vaccine works, as I explained in the mRNA, is continued. I hope you remember that. So, the Oxford vaccine requires 2 doses. The first dose is half the second dose. But there are data recently that show that this may not be the appropriate dosing. So it will depend on the different setting. The standard dose is actually 0.5 ml and not 0.25 ml. Now, let's look at the update on the AstraZeneca Oxford vaccine, because there is good news, but there is also bad news. And the latest data shows that a single dose and the influence of the timing of the booster dose on the immunogenicity and efficacy of the AstraZeneca vaccine is important. Remember, before the study, it was approved that it should be given 4 weeks apart. And the planned rollout of the vaccine in the UK involves giving 2 doses actually. And this time they studied it at giving it 12 weeks apart. The added value of this study showed that the updated efficacy results after an additional month of data collection from 332 cases of primary symptomatic COVID-19 patients explored the efficacy of a single dose vaccine for AstraZeneca Oxford. And they were able to demonstrate that in 17,177 seronegative trial participants, including those that were NAAT positive infections, these are those that had variants. They did a low dose and a standard dose and looked at the standard dose and standard. Remember the low dose, 0.25 and 0.5, and the standard dose groups, which is the 0.5 and 0.5. They showed that the overall efficacy was 66.7%. There was no hospitalization in the groups after the initial 21-day exclusion period. And the vaccine efficacy after a single dose from the day 22 to day 90 after vaccination was 76% if you gave the single dose, if you gave the standard dose from the get-go. So the anti-levels were actually maintained during the 3 months period. There was waning by day 90, which tells you that the standard dose group after the second dose, the efficacy was higher with a longer prime boost interval at 82.4% at the 12 plus weeks versus the 54.9% that was seen with the standard dose, but given 4 weeks apart. So we now know the data that you can give this at least 12 weeks apart. So after the first dose of the AstraZeneca, the second dose is recommended now to be given 12 weeks later or 3 months later, rather than giving it 4 weeks apart. The problem with this was that this was demonstrated in only the 18 to 55 years old patients. That's the reason probably why Switzerland did not support the data for AstraZeneca. Now on transmission, they also have new data to support transmission. The transmission studies per se were not included in the analysis, and swabs were obtained from volunteers every week in the UK study, regardless of the symptoms, so that they can allow assessment of the overall impact of the vaccine on the risk of infection, and thus a surrogate for the potential onward transmission. So that means if there was no impact of a vaccine on asymptomatic infection, it would be expected that an efficacious vaccine would simply convert severe cases to mild cases and mild cases to asymptomatic, with overall PCR positivity unchanged. So what they did was they measured the overall PCR positivity to assess the reduction in the burden of infection. The single standard dose reduced the PCR positivity by 67%. And after the second dose, the SD-SD schedule of 3 months apart or 12 weeks apart reduced the PCR positivity by 49.5% overall. And the data showed that it may have a substantial impact on transmission by reducing the number of infected individuals in the population. So this was the first study that showed a decline in the transmission. Unfortunately, there is a bad side. So there is backup study on the 12-week dosing interval. And the bad news is that it doesn't really work well in patients who have the variant, particularly the South Africa variant, the B.1.351. So South Africa actually stopped the use of the AstraZeneca vaccine, and additional studies also confirm that immunity from previous non-South African variant are bound to come down with another infection if they're exposed to the what we call South African escape variant. The efficacy study here for the South African variant using the AstraZeneca vaccine showed protection only of 10%. And the data is limited because this was done in younger individuals who went on to still have mild to moderate disease when compared to placebo. Next is the Johnson and Johnson. This has been approved, and they have announced already that it has met the primary endpoints of its interim analysis. They have applied for EUA. Overall, it has 57% efficacy in South Africa. When they were doing this, there was already circulating South African variants in the country where they were doing this, which was particularly South Africa. So that's not really a very bad efficacy considering the fact that it has 57% efficacy, and Johnson and Johnson's vaccine is a single dose. There is no added dose to this, although they're doing studies now to find out if they give the second dose, will it improve its endpoint efficacy. The other one that's interesting is Sputnik V. Some of the researchers worry that our immune systems can respond to an adenovirus vaccine by making antibodies against it. So it would render the second dose ineffective. So in order to avoid this, the Russian researchers used one type of adenovirus, which is AD26. And the second dose is AD5. So 3 weeks later, magkaibang vaccine ang ibibigay mo. One is adenovirus 26, the second one is adenovirus 5, which makes this a more difficult vaccine to develop kasi dapat yung unang dose in the clinical trials was AD26 and the second is AD5. So may color coding ka pa sa kahon niya. Does it work? Yep. It's safe and effective, and it showed at least 91.6% efficacy against the virus. This is the original virus, and it's completely protective against severe cases. The next one, the second to the last, are the protein-based vaccines. And this is exemplified by Novavax. No? This is unique in the sense that the modified spike gene here teaches the immune system to make antibodies to the spike protein. What they did was they inserted the gene into a baculovirus and allowed it to infect moth cells. The infected cells produce now spike proteins that spontaneously join together to form spikes as they do on the surface of the coronavirus. So it's the same method actually of growing and harvesting virus proteins for flu and the HPV vaccine or the vaccine against cervical cancer. So it's really not something new. So people get scared. These are new, these are new. No, it's not. It's not. They're old technologies. It's just that you don't know what the technologies are. So the harvested spike proteins now from the moth cells are assembled into nanoparticles. The nanoparticles mimic the molecular structure of COVID, but they mimic, but they cannot replicate or cause COVID. The vaccine is injected into our arms, and because each injection includes many spiked nanoparticles, the compound attracts now the immune cells to the site of the infection, and then the whole immune mechanism is then triggered again. So we go back to the antigen presenting proteins, the activated T helper cells combined with the B cells, breaks this up. All of the antibodies now splurge into our system, and these antibodies now spot the virus. And as in other vaccines, when you are vaccinated and you are later exposed to COVID, the antibodies lock onto the spike proteins and the coronavirus cannot enter our cells, and infection is thereby blocked. And in the same way, it can trigger activated killer T cells, and the infected cells put fragments of its spike protein on their surface, and they can destroy the virus before they even have a chance to produce new viruses. So Novavax vaccine is 2 doses. First dose and second dose is given um, 3 weeks apart. Now, what do the study results show? May result na ang Novavax. So this is another one that is in EUA application. People always tell me, isn't that the Indian vaccine? No, it's an American vaccine. It just so happens that the manufacturing site is in India because India has very beautiful manufacturing sites and they are more capacitated with that. Now, if the Philippines only had a manufacturing plant that could do vaccines, then we wouldn't have this headache and this problem. So it enrolled more than 15,000 participants, 18 to 84 years old. Look at the population that's more than 65. Almost 30% were more than 65 years old. And again, the primary and secondary endpoints. Remember, I talked about primary endpoint. Primary endpoint is whether you get sick or not. So of the primary endpoint, 56 in placebo got sick. They had 62 cases, 56 in placebo, and only 6 in patients that received the vaccine. So its overall vaccine efficacy is 89.3%. Now, this is one vaccine wherein it has data actually on the South African escape variant. Because at the time that they were doing this clinical trial, it was kind of late. So they started this clinical trial sometime in August and September. And that was when the South Africa escape variant was predominant and was circulating actually in South Africa. So they were recruiting patients who had the SA escape variant, and they were able to demonstrate even 60% efficacy. This is also the only vaccine that showed efficacy in patients who have HIV. So in patients who are positive for HIV, it has an efficacy of 49.4%. And the data suggests that the prior infection with COVID-19 may not completely protect against subsequent infection by the South Africa escape variant. However, the vaccination with this vaccine proved that it had significant protection. While they are still doing new construct against the emerging strains and selecting an ideal candidate, if you're looking at a vaccine that probably would address in actuality, not in the lab. Remember, Pfizer's vaccine says that they also are effective against the variants. That's based on lab. That's based on laboratory data, in vitro data yan. That is not based on clinical trial data. So based on in vitro data, it may work, but if you look at which of them had clinical trial endpoints, it was the Novavax or the South African vaccine, as the Novavax vaccine against the South African escape variants. Last is the inactivated coronavirus vaccine. I know I've been talking for almost an hour. I told them that I will stop in a few minutes. There are 3. One is Sinopharm, the other one is Sinovac, and the other one is Bharat-BioNtech. The name for Sinovac is CoronaVac, Bharat-BioNtech is Covaxin, while Sinopharm is Sinopharm or BBIPBP CorV. BBIBP CorV, all of them actually come from one major variant. No? From the Beijing Institute researchers, they obtained 3 variants of the coronavirus vaccine from patients in Chinese hospitals. And they picked one because it was able to multiply quickly in monkey kidney cells grown in bioreactor tanks. So how do you make an inactivated vaccine? Because this is actually one of the safest because it has the oldest technology. So what you do is with large stocks of the coronavirus, you douse it with a chemical called beta-propionolactone. The compound disables the coronavirus by bonding to their genes. The inactivated, so ang nangyayari, na-inactivate yung coronaviruses. They cannot replicate, but their proteins, including their spikes, these remain intact. and you draw off the inactivated viruses and mix them with a tiny amount of the adjuvant. Remember I talked about adjuvant in the beginning? The adjuvant is there to stimulate the immune system to boost its response to the vaccine. That's why when it comes to efficacy, the efficacy, you do not expect the inactivated vaccine to be as highly efficacious as the other platforms of the vaccine. And it is used for over a century. Jonas Salk was the first to use it when it created polio vaccine. So hepatitis A and rabies vaccines also use the same similar platform. And it has the same story on prompting the immune response. Remember that these are dead viruses already. They can be injected without causing COVID-19, same as polio when it is inactivated. And when it is inside the body, they are swallowed up by the antigen-presenting cell. And the whole mechanism of making antibodies with the B cells attaching to it, B cells locking on it, T helper cells activating it, is the whole process of how our body makes antibodies later on. Okay? So let's try to sum it up in the next 10 minutes because I think I've been talking too long. So the clinical, do the various efficacy values matter? Well, the clinical trial world versus the real world, yes, it matters. We'll notice that the real world is really different. Once the vaccines have been rolled out, there will still people that will still get sick. And all current data show only short-term efficacy as the vaccines in development have been barely out of gathering data, and many are still a combination of ongoing phase 1 and phase 2 clinical trials. None have been approved commercially, and none have long-term clinical efficacy data. So it's always a choice between COVID-19 first or any vaccine available. I always tell people that if there is one that is already available in the market and you have the opportunity to get it, would you want to get COVID-19 first, or would you want to wait for a vaccine that would be made available if there is no definite timeline for it? Of course, the other matter is on safety. And we need to remember that every stage of the clinical trial, safety is paramount. When significant safety issues are detected in any part of the clinical trial, they are not able to progress further. So what we do know is that all of the vaccines that have received emergency use authorization, that means a minimum clinical efficacy of at least 50% and a minimum length of safety follow-up of at least 2 months have been proven to have short-term safety data. The efficacy endpoints, the primary outcome is to determine whether it works or not and how well it works. So that's what we call vaccine efficacy. So the 3 endpoints are clinical efficacy, which means you don't get sick. The second is the prevention of severe disease, which means you don't die. And the third is herd immunity, which is very difficult to demonstrate. But it's an endpoint that matters. So the first two, on clinical efficacy and the prevention of severe disease, are important in itself. So to summarize, what do the data show? If you look at, I summarized it this way, to show you that this will, the vaccines that are available, the total number of doses, the vaccine efficacy. So this is in the prevention of clinical disease. What you want to look at is on the prevention of severe disease, which shows you that there are vaccines that have superior prevention of severe disease, including the Moderna, the Astra Zeneca, the Gamaleya, Sinovac, and Sinopharm. There is no data yet for Novavax on this endpoint. None of them have herd immunity. None of them are commercially available, but 3 of them have EUA approval in the country. So when you procure a vaccine for the country, you need to take into consideration its safety and efficacy, handling and storage issues. I mean, what will you use for certain areas? Can you store them at -20? Our ability to roll out the vaccines without any hitches, you should take into consideration wastage, particularly with the multi-dose vials. Kasi mahirap yung, and I'll explain that in a little while. No, it's really difficult to aspirate 0.3 ml without the needle that has a plunger. And that's not available in the Philippines. I've not seen the needle that has a plunger, because the plunger is important. Why? Because it pushes the small amount that is left in the needle into your arm. And then there is the prioritization, who should receive the vaccine first. There should be equity in procurement appropriation when the vaccine is rolled out. You must ensure that the patients complete the dosing requirements, because we know for a fact that a single dose does not work, and ensure that the programs are able to meet the numbers enough to result in herd immunity or herd protection. Now, this one I share with you from McKinsey and Company. Hazel shared this with me. And this is a table on the effective COVID-19 immunization strategy that addresses each of the components of vaccine adoption. I leave this with you. These are the key activities of vaccine adoption, from availability, administrability, accessibility, acceptability, affordability, and accountability. So we need to take into consideration all of these, particularly now during the pandemic when there is shortage of vaccines, and how to roll out a vaccine program correctly. Now, the last of the slides is on those with previous COVID-19 infections. We know now again that there is data that there is a robust spike antibody response from patients who have had a previous SARS-CoV-2 infection. And the preprint studies actually imply that those that have had a previous SARS-CoV-2 infection, they've come down with COVID-19. The question is, can we just give them one dose of the vaccine? And the studies show that yes, we can. And this probably during a time wherein there is shortage of the vaccine, this will probably be a time wherein these are patients that can wait before they get they get the first dose. They should still receive the vaccine. But before they get the first dose, if you've had COVID-19 just recently, you are one of those that can wait to get the vaccine later on. And this is just a guide by the DOH, to who and when to get the vaccine. So you must assess the vaccine recipient. So if they are within the age of less than 16, they have allergies to polyethylene glycol, or they have a severe allergic reaction after the first dose of the vaccine, these are patients that should not get the vaccine anymore. So do not vaccinate. Those, if you look at the other categories, everything else patients can get. No? If you have allergy to food and egg and medicine, yes, you can get the vaccine. You have a history of bleeding disorders, yes, you can get the vaccine. You use a different gauge and apply firm pressure, and so on and so forth. So patients wherein you can defer the vaccine for a later date include patients who are sick, patients with history of exposure to confirmed or suspected COVID in the last 2 weeks, patients who have been treated for COVID in the last 90 days, patients who have received convalescent plasma or monoclonal antibodies in the last 90 days, patients who are pregnant in the first trimester. You need to reschedule it. But if you are living in a highly endemic place, then please give it after the first trimester. And those who have received any vaccine in the last 2 weeks should be rescheduled. Those with autoimmune disease, HIV, cancer, underwent transplant, steroid treatment, bedridden, terminal illness, or those that are about to die, get a clearance first from your attending physician. I think this is the story, particularly in Norway, wherein there were a lot of deaths in those that were more than 90 years old, but these were terminally ill patients already who were about to die. Last slide. The last slide tells us that it is an ethical choice. This is from Pope Francis, who says that it is an ethical choice when we choose to get vaccines or not. It's a choice because when we're playing with health life, you're also playing with the lives of others. So even if, people like the Pope, people like Queen Elizabeth have signed up at their age and at their comorbids, uh, I think that that in itself speaks for how important it is for all of us to get a job. Because only if and when all of us who are eligible to get a job go and get one, can we really tame this pandemic. Thank you.
Thank you, Dr. Benji. That was extremely comprehensive and super informative. I think we learned a lot today. It was like an amazing lecture, actually. I'm sure I see a lot of questions here. Can I ask, you were talking about Pope Francis and Queen and all that signing up to do the vaccine, to take the vaccine. How do you convince, let's say, our household help or whatever, who don't want to take the vaccine, who have their own um, hesitations? How do we convince them that it's the thing to do?
Well, I can't start my video because the host does not want me to.
Oh, let me fix that for you. Sorry.
Okay, so everybody can see me. Yeah. Well, then we need to do a campaign. No? I think some of the issues on vaccine confidence is actually on trust. No? Trust on what they will get. Trust on how to that we need to impart to them that the first thing is to make sure that they understand what vaccines they're getting and how much protection they're actually getting from every vaccine, and why it takes uh, it takes a whole community to get the shots in order to bring down the number of cases. And I think another issue with them is the cost, because they're not assured whether it's something that they will get for free or that they will need to pay out of the pocket. So when something is given for free, we know for a fact that a lot of people are more than willing to get. And syempre, many of them are afraid because of the previous Dengbaksha and so on and so forth. Yeah.
Okay. Now let me
So
go
there's
to
an education campaign that we need to do for the lay.
and they've already conceptualized this campaign.
I hope so. The Department of Health should do that. But you see, if there is mistrust in government, I hate saying that, no? If there is mistrust in government, then whatever government says, people will not believe.
Yeah, there's a trust issue. That's true. Um, but when you were said about I noticed you said about Pfizer needing to be stored at -70, but you were saying that they've applied for FDA consideration for -15 to -25. That's our normal freezer.
No, that's not. That's not. That's still, that's a deep freezer. -20 is yung sa bangkay. No? If you, our bodies, I hate saying that, but that's the best description. -20 is still deep freezing. So -20 to -40 is you want to use a deep freezer. Your ordinary ref freezer will not bring you down to -20.
But in terms of logistics, how is it coming in here? It's being shipped frozen with cold storage.
Yes.
Already
I think
here.
Hazel knows more about the logistics with this rather than me. But all I know is that they must ship it the proper way, because any movement in the temperature, any disruption of the cold chain will make the vaccine less potent. In short, it will give you a false sense of security. It may not harm you, but it will not protect you as well.
Hazel, did you want to say something? If you have any inside information about the shipment?
Well, a lot of that is involves the packing. So there are several layers of the special packing, especially the -70, the ultra cool, and they also have indicators. So anytime the temperature is breached, it actually indicates. So you have to throw out the whole box. So that's why it's very, very sensitive. And not only is it in the warehousing, but it's also in the trucks, and then the receiving pharmacy or or
Or facility?
clinic should have the facility. That's why I've always been asking this about the government's plan of how this is going to be rolled out. After all, correct me if I'm wrong, Benji, vaccines are medicines. They're covered by the pharmacy law. And a pharmacy law says that vaccines can only be received by pharmacies, pharmacists and doctors, and then they have to have the capacity to store these vaccines at the right temperature. So that has to be watched out for.
So are we prepared for that?
I think we are. Benji?
I hope we are. Well, you see the news naman every day is practice, practice. We're just waiting for the vaccines to come. But if you see the news, practice here, practice there, practice here, practice there. I'm still waiting for the news that we're all out there at NAIA waving the Philippine flag for Philippine Airlines to touch down and give delivery of the vaccines. But we've not reached that point because I'm sure Malaysia had that the other day. They were all excited and that was in the news. But
When is the target date? Do you know?
OGK.
Okay, only God knows.
No, I think a lot of the Asian countries, our Asian ASEAN neighbors, were using the Chinese vaccines initially because that's also what they got initially, like Singapore and
Hong Kong started already, I think. Hong Kong and
Yeah, but
Singapore
whichever
started.
it is, like Bangladesh, for example, is Astra Zeneca, diba? And all of our neighboring like Laos is the same. It's an AstraZeneca vaccine. Myanmar, these are poorer countries than compared to the Philippines.
Okay, let's go through all the questions. There are quite a few questions here.
yeah, tough questions for Benji.
Yeah, sorry, Doc. Okay, if a healthy, uninfected subject got vaccinated, but before the second dose got sick with COVID-19, which is the new variant, can the individual still receive the second dose?
Depends on the vaccine that you got. If you got AstraZeneca, then the second dose would be useless. You want to get it, get it.
Okay.
But I
And
showed you already the efficacy.
yeah, what would you get though if you had a choice? Are you at liberty to choose those?
That was the same question that was asked to me last night sa TV Patrol. And my answer was anything that is available already. I do not want to wait to get COVID. It's a waiting game for
Okay,
all of us.
okay, good answer. Okay, Sinopharm and Novavax. I can't remember what you said. Do they have EUA here? Have
No,
they
they
applied?
don't have.
Not yet, no?
I think the one that's applying for Novavax is Unilab.
Ah, yes, yes, that's what I
Yeah,
heard.
so you can order from Unilab for Novavax.
Okay, with your technical explanation of how vaccines work, it is highly probable that mutation could result in the process of manufacturing
No,
vaccine. No?
no. The mutation will not occur in the process of a vaccine. Mutations occur because you are unable to control the infection. When there are more people that get sick, then mutations occur, variants occur. Why? Because the shift of the virus from individual to individual makes them want to survive that host. So vaccines will not do that.
How do we test if the vaccine worked? What is the next step after completing 2 doses? The vaccines seem not to work. It won't work if you get it, right? I mean,
If you don't get it, it's not going to work. Nobody has ever tested if the vaccines worked or not, no? I mean, when you get it, it's presumed that you will not come down with the infection when you are in the normal setting. Like I said, the real world is different from the clinical trial world, which means that in the clinical trial world, what we do is that after you received a certain dose of the vaccine, we take blood to determine if you develop antibodies against it. Kasi may baseline ka eh. So no antibodies in the beginning, and then later on, we keep taking blood to determine if you develop antibodies to it. But remember, it does not mean that just because you get the shot, you're not going to get infected anymore. You can, because it is not a perfect vaccine. It's like getting flu shots. No, you can get flu eventually, but it does not mean. But what happens is that your flu, the disease is milder.
Yes. So that's you're also protecting against getting a severe
Yes,
case.
actually, that's the end point that I wanted to drive at. You don't want to die. Never mind getting mild infections, right?
Okay. What
Of course, it's also
percentage?
important that you don't get infected.
Yeah.
But
Right
if
there.
I were to look
But
at
if
the
you
two parameters, I'd look at the parameter of no death.
Hmm. What percentage of the population should be vaccinated in order to have herd protection or to reasonably say we are safe to go out? You said this earlier, no? About
Yeah.
70%.
Yeah. Well, the calculation is around 70% depending on the vaccine that you are using.
In the meantime, while we haven't all been vaccinated yet, we'll still go on wearing masks and we'll
Yes.
all the
Even if you have
protocols.
been vaccinated, you will still go on wearing masks.
When do you envision some semblance of normal life returning, meaning a maskless existence?
Well, when a lot of people get immunized and when we see the number of cases down, I mean, in the Philippines is one of those wherein the cases have not moved and is actually going up compared to the world.
And now with the arcades and the cinemas opening at
I
20.
don't think the president has allowed that. I think in his statement last night, nobody will go back to MGCQ for a while.
So they're going to retract that whole
They
opening.
retracted it, I think, yesterday.
And the schools as well, right? He said.
Well, that's the other one that should be open. I mean, you're going to open something first, you open the school first because there are areas in the Philippines that have very low transmissibility. You know how children are, no? They're going crazy already in the house. Uh, I'm a pediatrician, so I know. I end up being a psychologist eventually.
That's true. You mentioned this already, no? The 100% efficacy against severe disease of Moderna, AstraZeneca, Gamaleya, Sinopharm, and Sinovac. Is it correct to say that these are so except against the South African
Yes,
variant?
correct.
What do
Yes,
we have
against the
for the
stand.
South African variant?
Nothing. Well, we have Novavax, which has 60% efficacy. That's
Oh,
the one of the better ones because it was actually done in a clinical trial. But the vaccine companies are rushing now, like Moderna and Pfizer, are rushing now to address the variants as well. So they're developing a booster vaccine against them. But the variants will change every time. So you'll have to keep racing against time against these variants.
eventually, will there be a vaccine that will cover all the variants, or it has to evolve the way the variants
I
evolve?
think it will, yeah, I think it will be a yearly thing. Once a
Like
year,
a
like just
booster
like
every
flu, we're
year.
going to get the shots.
And I think you mentioned this earlier, just to go back to it. Do you recommend taking a rapid antibody test after full
No,
dose of
no,
vaccine?
no. If you're going to do that, don't get the vaccine na lang. I mean,
There
you
are,
don't trust the vaccine, di ba?
yeah. Well, I guess people want to measure what they're
No, it's not a good measure of your con. That means you
okay.
should get it before you get the shot.
So you have a baseline.
Yes. How do you know if your antibodies are high because you got COVID, but you did not know that you got COVID because you were asymptomatic?
But if we're asymptomatic, if you said there's a lot of cases of asymptomatic people infected with COVID, how do we know? Is antibody the only test? Is the only that the only way we know?
Yeah. or that you swab everyone.
PCR.
You cannot be doing, yeah, you
No,
cannot
I
be
know,
swabbing
but
everyone.
in the beginning, everybody was so paranoid with that a little cough or a sore throat, and we think we had it and all that. Is it possible that we could have gotten it without knowing,
Yeah,
but
it's
then it doesn't
possible.
come up?
Yeah,
But will
it's
it always
possible.
come up with an antigen test or an antibody test?
The antigen test is useless. The antigen test is only useful when you are infected. But the antibody will tell you whether you got the disease or not in the last 3 months, 2 months, 3 months.
It's got a
So,
3-month window.
yeah,
But if you want to test antibody, you have to get the blood test,
yeah,
ideally, not the rapid antibody test. It's
yes,
not accurate.
yes.
Okay, and then there have been all there's talk going around that private individuals are bringing in Moderna vaccines, right? Um, so how is that going to happen without EUA approval in the Philippines as of yet?
If it arrives, it arrives, but it will not be given to you.
Oh,
You
okay.
cannot receive it. It will arrive. If it arrives, it arrives. Okay, but you cannot get it because there is no EUA. So unless there is an EUA approval, then you cannot get it.
So
Because
these things
whoever
are there, right?
sells it, yes,
Yeah,
if you administer it to somebody, tapos walang EUA, may nangyari sa taong yon, aba, yung
yeah.
buong kumpanya isasara.
Uh-huh.
Habulin lahat. Walang matitira
But
kay Rason.
okay. I wasn't trying to mention names, but
I'm just being blunt about it. You don't expect, you wait until there is an EUA approval as well. Kasi they come hand in hand. Di ba? So you wait. If it arrives, then it arrives. But if it's here already, then that's the good news. The second good news should be you wait for the EUA approval. You have to remember that all vaccines have expiration dates.
okay.
And as far as I know, the stability of all of these current vaccines are 6 months.
So if it
So
arrives
kung,
without the EUA, it will have to be stored. And after 6 months, if it hasn't received the permit, it'll have to be destroyed. Okay. Let's hope that the EUA comes soon.
somebody has to apply for it. That's the problem.
Let's see other questions here that you haven't actually answered already. Okay.
I already answered the
Yeah, you
antibody,
mentioned
antibody,
all.
antibody. No, 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 no.
There seems to be hesitation with the first-time technology mRNA vaccine among doctors, with reports of creating a more virulent strain as a complication. Is there any truth to this?
I know there's no truth to it, unfortunately. Um, really nothing much I can say regarding how much misinformation is getting circulated regarding that.
Yeah.
Diba?
Especially with, people seem to think that the RNA is gonna replicate, right?
Yeah, it's going to make you baog, it's
Yeah.
going to it's going to do a lot of things. Diba? It's going to make you probably weirder later on, or it might make you a zombie eventually. But
Or it will
let
be
me quote,
injected with microchips by
yeah,
Bill Gates.
let me
Microchip and
quote Fauci. Dr. Fauci said, it's ludicrous to think that it can genetically insinuate itself into our DNA.
okay.
It's not trained to do that.
And I'm assuming there's a high level of trust in what Fauci says here,
Yes.
right? Okay, there's still other questions here. Um, should a vaccine vial be broken in the process? Would this pose a threat to the nearby physical area?
No,
Not really, no.
no. They're killed. Wala yan.
If, say, you get a sloppy med tech who underdraws your shot from a 5-dose bottle, how does it affect efficacy?
What do you mean?
Maybe underdosed.
Yeah, you're underdosed instead of getting the zero point 0.3 ml.
Malas mo.
Yeah, but the efficacy is based on that amount, right?
Yes, the clinical trials show it should be at zero point 0.3. The
Yeah.
question is, was it was zero point 0.3 ml given to you? Kasi it may work, but
Yeah.
nobody has ever tried the lower doses.
Well, I suppose this is a possibility, especially if you're rushing the vaccine and you're rushing the rollout and you haven't trained your
Kaya
hopefully,
mas maganda ang single dose.
which is the Novavax, right?
No,
No,
which
the
is the
Sinovac.
Sinovac.
Sinovac. Oh, okay. Here's a political question. Why wouldn't the government give an EUA when it's urgent? Is that an
Because,
FDA thing?
no, I will give you an answer to that. Because you need someone to apply. You cannot give an EUA to somebody who has not applied anything.
Yeah.
Because you will blame the government naman na, why are you giving an EUA? Hindi naman pala siya nag-apply.
So it's an administrative thing, really.
Yes,
Yeah.
yes. You cannot give something to someone who does not ask for it.
Are all possible side effects from vaccine treatable? New York Times carried an article about a Filipina who developed a rare blood condition. Are Philippine hospitals able to treat such if it occurs?
Well, that Filipina that developed that rare blood condition, we do not know if it is vaccine related or not. You know, there are some things that we do not know. Like, for example, the timing of the administration of the vaccine and the occurrence of the disease. Because it's possible that you were already sick from the get-go. All right. But you just did not know.
Yeah.
I have people like that. I have people like that. I have patients like that, that they already had an underlying medical problem, but they do not know until some laboratory tests are done. For that Filipina may not have been having regular checkups. Her last checkup may have been a year ago. Diba? So let's not put one and one together just for the vaccines. It's really, really rare that you get um, I think the most important thing that you need to remember is all of the vaccines will cause some degree of pain. It will cause some degree of
Discomfort.
side effect, discomfort, yeah, your term. But the one that you're after is an anaphylactic reaction. Yung one wherein pagkabigay mo, bag patay, di ba? Or hindi makahinga. Yun ang more concerning issue. Hindi yung mga masakit eh. O, di ba? O yung sumakit yung ulo niya a day later pero nawala naman a day after. Or those that develop chills and a little fever 24 to 48 hours after the vaccine. But that's usually seen in the second dose of the mRNA vaccines because that means your immune system is alive, well, and kicking
But that
because it's
also happens
fighting
with
it
other
off.
vaccines, right? For other
Yeah,
illnesses, you can
yeah.
also get those reactions. Okay, now how do you feel about the immunization passport? People are talking about a vaccine passport being developed so that we could all travel knowing we've been vaccinated. That's a little bit of a contentious issue, right? Because
Yeah, I don't think that's within
privileged.
my my expertise anymore. That's more political than
Okay.
medical.
Now, if you get an allergic reaction after receiving the first dose and can no longer receive the second dose, are you still at least partially protected?
You
Well,
may
you,
be. Yeah,
yeah.
you may be. But I don't think it should be a contraindication to receiving the second dose unless that allergic reaction was one that was life-threatening. I go back to the point eh. Kasi if it's just an allergic reaction that you developed a rash or you developed some pruritus, then that shouldn't be a big deal. You can get the second shot.
How do we make sure that people get the second shot? That's the other thing, right? There's a kind of
Yes. Yeah, that's
a
why there
feeling.
should be that's what I presented Care of Hazel.
Yeah.
She shared that dapat may IT, may registry. Hindi pwedeng basta-basta na lang na we give the shot and then you sign it on a card. You need to input it as well so that people know what vaccine they got first and then that's when the second one is due. Somebody should contact them that the second one is due and when it is due and so on and so forth.
So
That's the practice that the Philippines should have. Hindi yung turok-turok on the side. Doon magaling na tayo. Pero yung practice doon sa registry at saka sa systems, dapat magawa natin.
okay, I think we have one final question. do you consider it ethical? For example, you're getting the vaccine, so prior to the administration, you check to make sure that the dosage is correct. I guess these are, just to be sure, because of the lack of, maybe there's not enough faith in the lack of experience in administering this vaccine. So, um,
So
should
what's
we
the
do that?
question?
No, I mean, can we check? I guess, I mean, it's within our rights, right, to check. Can you check
If
if it's 0.3 ml or?
yeah, yes, check. Yeah, you ask. Diba?
Okay, I think that's it for today. I think I'd like to thank everybody for joining us today. It was very interesting, extremely informative, but a little bit of information overdose. But you gave a really great overview of how the vaccine works and how our body responds to it. So hopefully, a year from now, we'll hopefully be, at least at a safer level than we are today. On behalf of Manila House, I'd like to thank CMI and Dr. Benji Ko for joining us today. And okay, wait, there's a follow-up question about the vaccine registry. You cannot be given a different vaccine, right? For the second dose.
No, no,
You gotta
no,
take the same one.
no.
Okay. Uh,
There's no interchangeability.
Okay, just a reminder, next week we have a couple of webinars coming up. We've got we have an art talk coming up on March 4, a talk about your mental and your mental well-being during quarantine on March 3. And we have a full program for March. Just stay tuned. We'll inform you in due course. And this talk has been recorded. It will be up on YouTube in a day or so in our Manila House YouTube channel. Before we close, Dr. Ko, what final words of encouragement of
Yeah, when you're eligible and the vaccine is available, make it our ethical and social duty to get a job. You're not only protecting yourself, you're protecting your whole family, your whole community, and this is the only way. Well, it may not be the perfect solution. It may not be the perfect solution, but it's the only way we can really move on from here, because a lot of us have suffered enough in terms of economy, socially, mentally, physically.
thanks. I
We
think
need
that
everybody in this together.
that pretty much sums it up. Yeah. Dr. Hazel, maybe final words from your perch over there in Europe, in Monte Carlo.
Well, it's the same thing as Benji said. We read, we always watch on the news how Israel and UK are doing it. They're already now laying out their plan to get out of the lockdown. UK said it's going to be now their last lockdown and they're opening up starting May. And it's something that we all are waiting for. We've been waiting for a year. I want to come home to Manila. So vaccination is really our only way out of this. And I hope that there's just so much misinformation and that's causing the vaccine hesitancy. So it is also all our responsibility to make sure that the right information gets out there. So thank you, Manila House, for helping us.
Thanks everyone for joining us today, and we hope to see you soon. Thanks, Dr. Benji and Dr. Hazel. Thank you to CMI. Okay,
Thank
bye
you.
everyone.
Bye. Bye, Hazel.
Right. Thank you.